Hi everyone. Thank you for listening of my paper research about the subject in vivo anti-inflammatory potential of Blantigo ovato on albino male mice, submitted by assistant lecturers Sajaa Ali Sharif and Mahmoud Nuruddin Hamza. We work in Department of Medical Techniques Laboratories, Al Farabi College, Baghdad, Iraq, and then Mahmoud work in Department of Molecular and Medical Biotechnology, College of Biotechnology, Al Nahrain University. Then we have a plan of this research, first with the introduction, then objectives and materials and methods for this paper, which discuss results of this work and then conclusions of this paper. Firstly, introduction. Medicinal plants have used as a wound healing remedy for countries in almost all regions of the world and in the treatment of a number of diseases apart from wound healing. Plantigo ovato is a species of flowering plant in the Plantagenesis family order Lamiaceae with around 265 species, which little plants typically have a thick tuft of basal leaves and long, leafless stalk bearing a terminal spike of the little blossoms. This plant is a dietary fiber acquired from the seed husk of Plantigo ovato. It is one of the most regularly utilized over-the-counter preparations worldwide, fundamentally used to control regulated bowel habits and clean the digestive tract. As of now, there is logically proof of the convenience of Plantigo ovato for several gastrointestinal sickness, namely constipations, diarrhea, irritable bowel syndrome, and ulcerative colitis. It can likewise diminish the danger of metabolic conditions by controlling glucose level in diabetic patients, raising lipid levels in hyperlipidemia, and supporting weight control. It is also known by several common names such as isabogol, psyllium husk, dietary fiber, Indian plantigo, plant psyllium, sandy plantinia, spogal seeds, etc. Plant distribution in all the world, for example, Spain, Tunisia, Algeria, Morocco, Georgia, Azerbaijan, China, India, Bahrain, Iraq, Jordan, and US. In view of this, the present studies focus on the knowledge of medicinal uses of the plants and the scientific investigation to confirm their medicinal values. Aim of this study, a study was investigating the burn healing effect of the aqueous extracted Plantigo ovato seeds. The extracted was used as an ointment to cure the wound in the preface اللي هي shortest at its possible time with minimal pain, discomfort and scarring to the patient is comparison to standard silverine ointment by Switzerland. Then discuss the materials and methods. Subjects, plant collection and identification from the local markets of the Baghdad recognized by Dr. Ibrahim Al-Juburi. And then preparation of the plant extracted was prepared according to 10 grams of plant powder was placed in a clean flask followed by 250 ml of distilled water, and then it was placed in the shaking incubator at a temperature 28 centigrade for a period of 30 minutes. Then filtrated using medical gas, and then the filtrate was distributed on plates. Dried this place in the oven at 37 centigrade until the water evaporated completely to obtain the dry powder for the aqueous extracted. This placed in refrigerator at 4 centigrade until use. We have a laboratory animals using Ilmas it was albino male mice. The age of these mice between 8 and 10 weeks. not less than or more than, because it cannot handle the abnormal condition. And the weight of this mice, 23 to 27 grains. The animals was distributed in two groups, which each group kept in a separate plastic cage and offered maintain at room temperature and had free access to food and water. Then we have experimental design. Three groups were tested, which each group contained three animals. The total number of animals in this experiment was 9 mice. Then we have immunological parameters using Gilbert's skin model in mice. In order to assess the burn healing activity of Plantigo ovata, three mice groups were tested. And mice hair was removed hair and the flame was used to induce the burns of the skin. Then calculated recovery days by determining number of days required to heal the wound. Here, the animal groups were categorized as the following. Three groups were tested. Each group contained three animals parameter, total number 9 mice. The details of these groups summarized in table 1. Group 1, mice without any treatment, which is considered as a negative control. Group 2, mice with administration with the Plantigo ovata اللي هو aqueous extracted. Group 3 mice were administered with commercial cream of silverine Switzerland. Total number of mice in this stage was 9 mice. Results and discussion. The outcome show aqueous extracted had although expanded the percent wound decrease, promoting improved injury repeating in this table. Group 1 without any treatment, the period of recovery needed 24 days. Treatment with the Blantigo ovata al-azabagol needed 15 days to full recovery, while the treatment with the silverine Switzerland required 18 days to full recovery. The crushed form of leaves are used in healing of chronic wounds. Recent ethnopharmaceutical studies show that the Plantigo ovata is used in many parts of the world in the treatment of number of diseases where Plantigo leaves and seeds contain carbohydrate, lipids, vitamins, caffeic acid, derivatives, flavonoid, aridol glycoside, alkaloid, and other organic substances lead to its diverse medicinal properties. Each of the constituents has unique medicinal property. Plantigo ovata isabogol has also tested for wound healing properties by assaying the proliferation and migration of oral epithelial cells in vitro, and the results exhibit the extracted of Plantigo ovata have a benefit effects on proliferation of epithelial cells, suggesting it is wound healing. Conclusions. We have one conclusion is that Plantigo ovata and acts as treatment for burns by decreasing the day required to recovery due to the presence of chemical constituents of the plants, due to have many materials, flavonoid, alkaloids, and etc. Thank you for your listening.